The overall goal of the following experiment is to test the effects of plant-induced soil heterogeneity on plant populations and individual responses related to community assembly. This is achieved by collecting soil from individuals of two focal plant species in the field to represent different patch types of plant-influenced soils. As a second step, soils from each focal species are placed in separate patches within a pot, or mixed throughout the pot, creating a heterogeneous or homogeneous soil treatment, respectively. Next, the focal species are planted in each soil patch type to assess plant performance in heterogeneous and homogeneous soils. Ultimately, the effect of plant-induced soil heterogeneity on plant performance can be assessed by analysis of plant traits, plant growth, germination, or survival.
This method will help us to answer basic questions in community ecology, such as what is the role of heterogeneity in governing community assembly and plant diversity? Or what's the role of plant-soil feedbacks in governing coexistence in the field? Helping to demonstrate these techniques today will be undergraduates from my lab, Jacob Hooks and Angela Kazauka, and we're off to the field.
To produce hetero and homogeneous field soil treatments, begin by collecting field soil from marked individuals of the two focal species of interest in replicate batches to avoid pseudo-replication. Using an appropriately labeled sterile shovel and gloves, next, dig up the complete root zone.